So, oops, I haven't translated the title, but today we're going to talk about a real research project. And so we'll try to apply some of the things we have discussed before in the frame of our conceptualization of how a research project is thought of, planned and carried out, to a real one, and see how those notions, those ideas apply to an actual research project. So first of all, a little background to understand the research project we're going to talk about. The laboratory who did this study, is concerned with the relationship between the control of the cell cycle and terminal differentiation. And they are interested in two different aspects of the post-mitotic state, to say it in short. First of all, they would like to know what are the molecular determinants of the post-mitotic state, which is incompletely unknown. So we don't really exactly know why and how a cell decides not to proliferate anymore. And this is a basic question. The same laboratory is also interested in reactivating the cell cycle, reactivating proliferation in terminally differentiated cells for regenerative medicine purposes. If we had the ability to induce proliferation of cells such as neurons, cardiomyocytes, cells of endocrine glands in a controlled, reversible, safe fashion, that would be invaluable for regenerative medicine. So the system the laboratory has chosen to work with is a model system, and it's the skeletal muscle as recapitulated in vitro. So in vitro, you can grow myoblasts, you can expand them to the number you wish. Then you can easily induce differentiation of these myoblasts into myocytes, mononuclear cells, which are nonetheless already terminally differentiated. And in fact, in the skeletal muscle, usually exit from the cell cycle is a requirement, is a prerequisite in order to differentiate. Eventually, the myocyte will fuse into myotubes, which are syncytial structures, multinucleated. So, this is the system they work with. They assume, and they have in previous literature demonstrated in several cases, that this is a typical example of terminal differentiation, that other post-mitotic cell type also respond in similar ways as the myotubes to a variety of stimuli. So the story here starts from the knowledge acquired by this laboratory that terminally differentiated cells can be reactivated by forced expression of two cell cycle regulators, cyclin D1 and CDK4. Of course, you know that cyclins are the regulators of CDKs. Do you?
I think we show it in the previous lecture.
Yeah, but you haven't studied the regulation.
No.
Okay, so something we have to say something more then. So the cell cycle is regulated at a very fundamental level by the activities of a number of kinases called cyclin-dependent kinases. They are called cyclin-dependent because they are only functional, they only have kinase activity when they're bound by their cognate cyclin. Not all cyclins can bind all CDKs, so there are associations that exist in nature and others that don't. So the authors found that forced co-expression of cyclin D1 and CDK4 can reactivate the cell cycle in terminally differentiated myotubes. And so they devised two recombinant adenoviruses, one carrying cyclin D1 and the other CDK4. The reason why they chose adenoviruses is, first of all, because they infect efficiently cells in culture, and they are, and they also have a large number of hosts, meaning they can infect a variety of cells, so it is relatively easy to export the results obtained with myotubes to other terminally differentiated cell types. So what they found was that by infecting myotubes with these cells, they could obtain a very high overexpression of both cyclin D1 and CDK4. These are Western blots, and what you see here is control myotubes, with relatively small amounts of both cyclin and CDK, infected myotubes with very large amounts of both proteins, and for comparison purposes, the amounts of cyclin D1 and CDK4 expressed by myoblasts, freely proliferating myoblasts. Okay, so this huge overexpression of cyclin D1 and CDK4 drives myotubes into the cell cycle, and these are myotubes stained in red for myosin heavy chain, a marker of myotube differentiation, muscle differentiation, and in green for BUDR incorporation, so again, as we have said other times, demonstrating that they have synthesized DNA. These are control myotubes infected with empty viruses that, of course, do not re-enter the cell cycle, do not synthesize DNA. And the technique shown here is quite efficient in that in this particular experiment, more than 70% of the cells re-entered the cell cycle, and they did so in a fairly synchronous way, meaning that before 24 hours from infection at time 0, little happens in terms of BUDR incorporation. Then altogether, within a few hours, all the cells that will be eventually reactivated enter S phase, and then the number stops increasing because all the cells that could re-enter have done so. So this is advantageous when cells behave synchronously, because you can treat the whole culture as if it were a single cell. Whatever happens in a single cell here is reflected in the whole culture. Working with a whole culture, of course, is much easier than dealing with a single cell. All right, so this is in the background. The last information that we need to provide you is that the authors now have a question in their minds, that is, why is it necessary to overexpress at such high levels cyclin D1 and CDK4 to obtain re-entry into the cell cycle of myotubes? It appeared to them that it was really an artifactual stimulation that had little to do with the physiology of the cells, except, and this is the last piece of background information, that they measured the kinase activity associated with cyclin D1 CDK4 complexes, these active kinase. And they found, to their surprise, that the amount of such activity measured in myotubes reactivated by double infection was quite comparable, practically identical to that found in freely proliferating myoblasts. That implied that whatever the amounts necessary to obtain this kinase activity, the kinase activity sufficient to reactivate the myotubes was entirely physiological. And I'll add that what drives the cell cycle is not the kinase in itself, but its activity. So basically, what they had found was that it is sufficient to reconstitute normal amounts of CDK4 activity, which is absent in control molecules, to reactivate the cell cycle in these cells. Now, we start with the project, and the usual 4 levels. We have the question, the experimental approach, the design of the experiment, then of course the experiments are carried out, and finally we have to interpret the results. The usual process. Per favore parlate piano, grazie. Good, so first of all the question. Well, we can suppose that the big question these authors have in mind is, there's the question there, it's okay. Why terminally differentiated cells refuse to proliferate? Well, this is of course an all-important question, but it is vaguely formulated. It's too wide. As we mentioned when we talked about the question, we need to focus the question in order to make it addressable, answerable with the scientific method. Not that this is an unanswerable question, but probably it will take a number, we don't know how many, a number of steps, a number of sectorial understandings before we can compose a whole picture. So, the authors needed, first of all, a better question to start with. And they chose to focus on a rather obvious question that stemmed directly from the results I've shown you. That is, they asked themselves, why is it necessary a very high expression of cyclin D1 and CDK4 to attain physiological levels of kinase activity, of CDK4 kinase activity in myotubes? There was a sort of apparent contradiction between the huge overexpression of the two molecules and the normal amounts, normal levels of kinase activity. So, now this is a tractable question, and I'll ask you to propose ways, to propose approaches to answer this question. Why would you think that there might be such a need for very high levels of the two molecules to obtain normal levels of kinase activity? Of course, you don't know about the cell cycle, so that is a problem, but you can still think in abstract terms.
There are some repressor while the cells are differentiated, terminally differentiated, that even if you add a normal amount of the cyclin D1 and CDK4, they don't let them have their normal activity.
Karim agrees with this idea.
Yeah, I was thinking the same. In particular, maybe after the differentiation, we can have a decoy model that shift the activity of CDK4. Something like that.
Excellent. Good answers. Yes, that is in fact what the authors thought. And so the question shifted, basically the question was there, and they shifted to think of the experimental approach. This is the question. So, they needed now, it must have been presentation, but anyway. Then is now a model that allows them to propose an approach, to design an approach. And so basically, you've just proposed a rough model. You both said there is an excess of inhibitors of the kinase or the kinase activity, which prevents normal levels of the cyclin and CDK from producing an activity. And that's what they thought. But then the question is, okay, what do we do then? If that's the model, how do we approach our original question?
Trying to follow the activity of CDK4 from the start of the sampling itself, and show with what who complex and protein they interact also.
I'm not sure I understand. Can you rephrase, please?
Yeah. I think that we can follow the activity of the kinases during all the cell cycling, and then if we can mark CDK4, probably we can find also the other reagent that we can have in during the cell differentiation.
Okay, so your thinking is, in myoblast there is an active CDK4,
Yeah.
but during differentiation probably something else happens, something else gets expressed or overexpressed,
Yeah.
which blocks the activity of CDK4. And how do we find it?
I can find the CoI protein that can
CoIP, you say?
no, the CoI protein. I don't know how.
Oh, the CoI protein.
The CoI,
yeah. Okay.
And show if something else can react with the CDK4.
All right.
Probably.
What do you think?
Yeah, cause yeah, you said it while like using CDK4 as a trap to co-immunoprecipitate with the inhibitor. So like
I can do it in vivo.
Well, we'll find out how to do it.
Okay.
so we could at least see all the things that interact with CDK4, and then try to overexpress or repress the thing that interacts with CDK4 to find the right one, that is the one that stops its activity in a differentiated cell. Something like that.
You agree with that?
Yeah, I was thinking that it's not possible to do an co-immunoprecipitation in vivo.
You don't have to do it in vivo. You can do it
You
on a
kill
cell
the
lysate.
cell.
Yes, you kill the cells and then do the co-immunoprecipitation.
So
You kill.
So you question whether the approach is feasible. That's a good
I can
question.
see if
First of all, before designing an approach, you have to determine whether it is feasible or not. Okay, let's see. First of all, excellent thoughts. In fact, I should suggest the authors to hire you in their laboratory because you're pretty good. So, here is the model the authors devised. In a non-terminally differentiated cell, of which this is the nucleus and this is the outer membrane, the cell membrane, when a growth factor binds its receptor, it activates, remember this is a schematic, this is a very simple model, so it's highly simplified. when the growth factor binds its receptor, it activates a cascade, a signaling cascade that goes to the nucleus and induces the expression of a cyclin D. There's a family of cyclin Ds, there are three of them, cyclin D1, D2, and D3. It's undetermined which one is relevant in this case, but it doesn't matter because they behave much in the same way. CDK4 is already expressed, even in terminally differentiated cells, so there is no need to activate its expression. And in non-terminally differentiated cells, a complex is formed which has kinase activity which drives the cell cycle. Now, the authors agree with you and think that probably helped by the literature. You guessed it from scratch. You didn't know about the cell cycle, and you guessed that there might be an inhibitor. Indeed, the literature at the time suggested that there were, in fact, plenty of inhibitors in terminally differentiated cells, expressed at much higher levels than in non-terminally differentiated cells. And, but the literature was confused. It was early days in terms of inhibitors in the cell cycle, so it wasn't clear which ones were expressed in which cells and at what time during differentiation. So the authors completed this model by saying, well, presumably in terminally differentiated post-metabolic cells, there is one or more inhibitors which bind either CDK4 or cyclin D or the whole complex depending on the inhibitor. And this prevents the myotubes from responding to growth factors. They do not proliferate in response to growth factors. Now, this model makes predictions. So what useful prediction does it make?
That we have an inhibitor that stops the growth factor activity
And
CDK4 activity.
That
yes,
binds, so we can.
you can do the cranial
yeah.
precipitation.
Yeah.
Yes, but
Or if no, I don't know if we can do an extract of CDK,
yes, we can.
and if we know the structure, we should know the weight of the complex.
Close enough.
Yeah.
But the question now is at the biological level. Does this model suggest anything to you? What kind of manipulation could we subject the myotubes to in order to activate the cell cycle in the myotubes? It's pretty easy.
Repressing the inhibitor.
Repressing
Yeah.
the inhibitors. So the authors thought if we could identify this inhibitor or inhibitors, then you know which and how many were there, and we could eliminate it, we would reconstitute the ability of myotubes to respond to growth factors, which was the holy grail of the field at the time of this study. And so, even though there were quite a few clues in the literature as to what inhibitors might be involved, they decided to take an unbiased approach. Remember what the unbiased approach is without preconception, without hypothesis. The general hypothesis is there is an inhibitor, but we don't try to guess which one it is. We can even conceive that it is a yet unknown inhibitor. So we are not targeting specific inhibitors. We want to know what inhibitor is associated with CDK4, as you suggested. So to answer Karim's question, can you do that? Yes, you can. And the way is this. So you now have a model that allows you to devise an approach. So we need to identify the presumed inhibitor, which is also an interactor of CDK4, and we'll do it through immunoprecipitation. Okay, I'll remind you, although you know, because otherwise you wouldn't have guessed this approach, that in an immunoprecipitation you have a number of molecules, usually proteins, and that some of them interact with one another. For example, here the blue one interacts with the red one. And if you have an antibody against, for example, the red one, and you use it to immunoprecipitate the red molecules with appropriate beads, we have seen this, you would also immunoprecipitate all the interactors that have a strong enough interaction not to be broken during the procedure. So,
Brainstorm.
now laboratory brainstorming, exactly, discussion on how to proceed, and so this is the design of the experiment. How do we do this? How many cells do we need? What are we going to precipitate? Do we have an antibody to do so? How efficient is our procedure? What do we do next? And then, through a lot of discussion, they come up with this design. We take myotubes. This time, this graphic here represents a myotube. And we overexpress cyclin D1 and CDK4 just below the threshold necessary to reactivate the myotubes. The authors didn't really want to reactivate the myotubes because that would induce a number of changes in the cellular environment. They just wanted, hopefully, to capture all of the available inhibitor in order to efficiently precipitate it and identify it. They would then simply lyse the cells, so we now have a watery solution of thousands of different proteins. They would precipitate them with an anti-CDK4 antibody, and they will separate the precipitated proteins on a gel. Eventually, once they've found proteins, bands on the gel that look interesting, they will identify them using protein mass spectrometry. So this is the actual experiment that the authors ran. As you can see, there are two lanes here. One is the anti-CDK4 immunoprecipitation, and here is a control. Of course, an experiment needs a negative control, we know that. This is not actually an experiment. This is, you would classify it as an observation, because we are only taking note of what exists in the cell. You are not trying to modify the cellular biochemistry. You need this control lane here because immunoprecipitation is never clean. There are always proteins that precipitate non-specifically, either because they bind beads or because they bind non-specifically the antibodies, or because they are simply sticky, as we say. They tend to attach to anything. A typical one is beta-actin, which is very abundant in a cell and it sticks to anything. You always find beta-actin in whatever you precipitate. This is beta-actin.
And the control is just CDK4? Or
No,
is another...
it's an identical immunoprecipitation with an irrelevant antibody.
Okay.
An antibody that doesn't recognize anything in the cell. It's against a viral protein, so
Okay.
this protein does not exist in the cell.
So in this case, the interesting band are the ones that are, it's
Sure.
like the one in the down, that one.
About the autism,
Yes, the interesting bands are those that are present in the true immunoprecipitation and not in the control. The common bands are presumably background, non-specific precipitation. Right. So the authors, yeah.
No.
No.
The authors identified the protein content of some of these bands, such as this one, that contains all three cyclin Bs, cyclin B1, B2, and B3, plus CDK4, because they have similar molecular weights, they co-migrate in the same band. Here they found PCNA, which is interesting because it's a factor involved in DNA replication. But the most important band was this one, which contained P21, which was even at the time a known cell cycle inhibitor, a known inhibitor of cyclin-dependent kinases, and the only inhibitor identified in this immunoprecipitation. So the authors did the obvious next experiment, which is
Mark
suppress.
Karim first
trying to mark P21
to do what?
to show in vivo if P21 get association with
Right, they could try to confirm this result, because immunoprecipitations are not necessarily a final proof of two proteins associating in vivo. They might, for example, just to make one of the many possible examples of an artifact, they might segregate in different compartments of the cells. They might never see each other, and yet when you lyse the cells, they come together and bind, but they don't in nature. So you're right. If they wanted to make sure that P21 did bind CDK4, they needed to do further experiments. But in this case, they were lucky because it had already been shown that P21 binds anything. It binds all cyclins, all CDKs, and all possible complexes between cyclins and CDKs. So it's a universal inhibitor. And you were suggesting?
Yeah, that's what we designed the experiment, like find it, suppress it, and see if it
exactly.
starts.
So let's follow up. We found it. Let's try and suppress it. Here is the suppression, an siRNA against P21. This is a control siRNA. So this is the amount of P21. These are Western blots. The amount of P21 found in unperturbed, in control myotubes, and this is what you get once you suppress it. So there is a very significant suppression of P21, and to the author's amazement, as soon as they suppress P21, the myotubes re-enter the cell cycle straight away. Now, it is important to tell you why the authors were so surprised. After all, this is the result they were actually longing for, and yet when it came up, they were surprised. Why? Because if you remember the model said if you can remove the inhibitor, you would reconstitute the ability of the myotubes to respond to growth factors. But the authors had not added any growth factors in this culture. So the myotubes re-enter the cell cycle even in the absence of added growth factors, which needed a different explanation. The model was no longer capable of explaining the observations. And so, what did we say about the models? That they are extreme simplifications of reality, that you should use them as long as they're useful, but once they depart from reality, once they depart from the observation, you should abandon them right away. Clearly, the model could not explain this result because, in principle, actually in theory, growth factors here would be needed for two reasons. First, because cyclin Bs are expressed when growth factors are around. And so in the absence of growth factors, there should be little cyclin B in these myotubes. Second, because growth factors are also needed to greatly facilitate cyclin CDK complex formation, And so even if there were cyclins and CDKs in these myotubes, in the absence of growth factors, complex formation would be very inefficient. And yet, the evidence was that the myotubes re-enter the cell cycle nonetheless. So in fact, the authors were so surprised by this result that they repeated the experiment another couple of times just to make sure that they didn't mess up with something that was just a spurious result, but the result came again and again, came up the same. And so, at the very least, they satisfied the basal level of redundancy. Do the experiment again and again, and if you get the same result, well, you tend to believe it. And once they decided they believed the result, they needed to explain it. And so this is another lesson that we will apply to this project. Because what they did was they went back to the immunoprecipitation. I mentioned, and probably didn't stress enough, that usually experiments tell you much more than what you're looking for. If you're lucky and the experiment works, you'll find what you were looking for. But the experiment has a variety of results, shows a variety of different things that you don't care about, that you ignore. But if you need extra explanation, go back to the experiment and see whether any clues are in there. And in fact, in this case, the experiment was trying to tell the authors something, but they didn't listen in the beginning. But fortunately, they went back and listened more intently. What I noticed on going back was that, among other things, there was cyclin D3 here. Now, the authors had overexpressed in the myotubes cyclin D1, so this was endogenous cyclin D3. This implied that, at least in these experimental conditions, under moderate overexpression of cyclin D1 and CDK4, the myotubes contain cyclin D3 CDK4 complexes, because here we had immunoprecipitated, they had immunoprecipitated with an anti-CDK4 antibody, and yet cyclin D3 came down. So the authors remembered data in the literature saying that myotubes contain multi-protein complexes that, among other proteins, contain cyclin D3, CDK4, and P21. Now, the authors are devising a new model, so they don't even bother to draw the other proteins. They think they're irrelevant. What the authors think here is that myotubes do already possess preformed cyclin D3 CDK4 complexes. And so they are ready to work, they are ready to activate it, except they are kept in check by P21. What happens, according to this model, what happens when they remove P21 is that they immediately activate these complexes. And thus, they don't need growth factors. They don't need growth factors to express cyclin D3 because it's already there. And they don't need growth factors to facilitate the assembly of complexes because they're already assembled. That would explain everything. But now, for this model to be useful, it must make predictions that we can test. What might be the prediction?
Looking for the amount of cyclin D3.
Yeah, and then?
And then follow if we have a correlation between the concentration and the activity of the complex.
Okay, the authors indeed have done something like that. But now we are talking about, again, a biological prediction. In what conditions? What conditions would allow you to validate this model? Well,
probably in the time during the cell cycle. We have
well, remember, in the immunoprecipitation there was no cell cycle. The myotubes were not reactive.
not okay, not different.
So here you have a model which says, if we remove P21, these two molecules drive the cell cycle.
So
How would you validate it?
if in not differentiated cell we express P21, they should stop
They do. It was already done.
It was already.
I think we have to focus on the activity of D3. I think.
We
Remember that the activity of D3 is
Depends.
simply activating CDK4.
Okay.
So we have to prove that it's true that D3 is already there and it's not because
Think in functional terms, in biological terms, not in biochemical. So do not think of the molecules. Think, if the mechanism activating the cell cycle is this one, depicted here, how would you support your hypothesis?
Inducing overexpression of the factors D1, D3, all of them, and show if we have more activity of the complex with CDK4.
Yeah, but the
they
authors have
did
already done it.
this
That's
time.
what they do to reactivate the cell cycle in myotubes, right?
Also with the
overexpressing.
inhibition of P21.
I'm sorry?
Also with after the inhibition of P21.
No, they haven't done that yet. But they will?
Yeah.
And I was thinking of something.
So the question is how observe that D3 and the complex inducing the cell cycle.
Yes, yes.
This is the question. Okay.
But again, think in functional terms, not biochemical. Not a biochemical approach. So you don't want to measure molecules or activities and such. You want to look at myotubes. If it is true that these molecules
inducing another inhibition of the D3 factors, and if I stop also P21 and if I inhibit P21 and cyclin D3, I should lose the capacity of the purify and doing the cell
what do
cycle.
you think?
Yeah. Makes sense if you
Should have not the
but when you want to stop in
before.
differentiation,
Yeah.
while there
Or even after differentiation.
If
Yeah,
I
yeah,
stop
I'm
it,
thinking-
I know that depends from D3.
Exactly. So that's in fact what the authors did. They reasoned that if they remove P21 alone, that would drive the myotubes into the cell cycle. But if they remove P21 and cyclin D3,
You should lose.
they should not go into the cell cycle. If they remove P21 and CDK4, they should also not go into the cell cycle, assuming that these are the only molecules involved in driving the cell cycle in the myotubes in this setting. So if they are necessary to drive the cell cycle and you remove them, you don't get the cell cycle.
Yeah.
And that's the experiment the authors did. It's a bit complex, but we have time. I will try to illustrate it and make it as simple as possible. So these are Western blots, and these are kinase activity assay. showing P20, the proteins indicated here. Focus on P21, cyclin D3, and CDK4. So, in myoblasts, there is little P21, and there is a good amount of cyclin D3 and CDK4. All right, so then the authors suppress. So this is a control lane. They mock interfere with P21 and mock interfere with cyclin D3. But this is basically a negative control. The cells were not essentially altered, and indeed there was no kinase activity associated with CDK4. Good. Now here they suppress P21, and again they mock interfere, control adenovirus. They mock interfere with cyclin D3. So the amounts of cyclin D3 basically don't vary, but P21 goes down strongly.
It's the first experiment.
Exactly, it's the previous experiment. And indeed you get activity, and we'll see what the myotubes do in response to this activity. Here, this is a bit of a complication. I will tell you the experiment exactly as it was carried out. They suppress P21, so again, very low levels, much lower than in control myotubes, but they also express a dominant negative CDK4. Now, I'm sure you don't know what's a dominant negative. It was a concept very interesting before we devised different easy ways to interfere with molecules. When these methods did not exist, what people did to interfere, say in this case with a kinase, was using a dominant negative. What is a dominant negative of an enzyme? Is an enzyme, is the same enzyme with a mutation that inhibits the enzymatic activity. So this dominant negative will compete for the substrate with the endogenous wild-type enzyme, and if it is overexpressed, it will prevent the enzyme from working. It will take away the substrate. So basically, this experiment is equivalent to suppressing P21 and CDK4. And in this case, there is no kinase activity. And then finally, if you suppress P21 and cyclin D3, they're both suppressed, there they are. They have no activity, and this is already very encouraging, so this is in accordance with the model. But what's important at the functional level is how the myotubes respond, and the answer is very, very clean. If you interfere with control siRNA, no myotube, of course, re-enters the cell cycle. If you interfere with P21 alone, in this particular experiment you get 68% of the myotubes to re-enter the cell cycle. But if you interfere with P21 together with cyclin D3, this number drops to 0. If you interfere with P21 and inhibit CDK4, again the number drops to 0. So, altogether, this experiment fully validates the model and tells you that the model is sustained. Doesn't mean it is true. It means it's okay, we can believe it until we get proof that something different is going on. It also tells you something that was implied in the model. I mentioned it. It tells you that there is no replacement for cyclin D3 and CDK4. No other CDKs or cyclins can substitute for them. If we remove them, they're gone. There is no re-entry into the cell cycle. So, in this particular setting, cyclin D3 and CDK4 are the sole molecules responsible for driving the cell cycle in the myotubes. And this is a conclusion the authors are happy with. So now we are again at the level of the interpretation of the results. Remember, we have talked about data, results, and conclusions in the last lesson. So what are the data here? They are these bands.
Yeah.
They are these numerical results. So these are the data. The results are the immediate consequences of these data, which are just stated. The immediate consequences are that the model is validated and that cyclin D3 and CDK4 are the sole responsible, the sole molecules responsible for cell cycle reentry in myotubes. But now we come to the conclusions, which is a wider interpretation. Again, nobody cares if my myotubes in my laboratory, in my conditions, do something strange. They care about the physiology and the possible alterations of this physiology in myotubes. So we need to generalize or extend the meaning of our results. And so, oops. I thought I had translated this line. Perhaps this is just an old copy. Never mind. So, what kind of conclusion would you draw from these experiments? If you were to write the discussion of the paper, what would you write? Okay, so now we know that cyclin D3 and CDK4 are responsible for blah blah. What else?
Would it be too much to say that cyclin D3 and the other molecule are for all the terminally differentiated cell?
At least you might speculate. You might say, well, it is possible.
That in all
The typical phrase, the typical sentence in discussion is, future experiments will tell whether cyclin D3 and CDK4 are responsible for reentry in all possible or larger.
I would say that to look more.
Right, so you already suggest something, although you have no way to prove it. But at least you suggest a way to generalize your results. What else? There are many ways to interpret the experiment. So I will show you one conclusion that the authors drew. It's not the only one in the paper. It will be just an example. But you can come up with conclusions that the authors never even thought of. So feel free to say whatever comes in your mind based on these experiments.
Starting from the question, I think that we can generalize saying something like the myotubes can do the differentiation if they lost the inhibitor P21, or cannot do
that's
cycle.
interesting. Oh, that's interesting. Yeah. So you're saying if you suppress P21, if they lose P21 somehow,
we can.
they de-differentiate. Is that what you're saying?
Yeah.
That's interesting.
Or that we can say also that we have no differentiation of cell cycle in myotubes because we have the inhibition of P21.
Excellent.
So from those two affirmation, we can start to a new project to.
Exactly. So this is not really a conclusion of this work here, because it would be too far-fetched, too distant from, not supported by the data. And yet, it's a good question for the future. You are suggesting what next we might investigate. Perfect. Okay. Indeed, I can tell you that if you reactivate the myotubes in this way, they do de-differentiate, at least in part. You were going to say something, perhaps?
No, I was surprised, and it's but the conclusion, it's the result is like
clear. The result is clear. It speaks by itself.
yeah.
They have a good app, so
A new model for
Okay, one thing the authors speculate a little about is that it is as if the myotubes kept their engine on.
Ah,
That is,
that's also
although
true.
they are destined never to be reactivated in nature, they nonetheless keep expressing all the necessary machinery to reactivate the cell cycle. Why? Perhaps, and because of that,
Yeah.
they need to push on the brakes continuously, because otherwise they would reactivate their own cell cycle, which is not good. And so, surprisingly, they waste energy to produce these proteins and more energy to produce inhibitor of these proteins. So, first of all, people assumed that the post-mitotic state was merely the result of the absence of the cell cycle machinery. Everything is shut down, nothing is expressed, and so the cell cycle is dead. But it is not so. The machinery is there, can be reactivated. In fact, it needs to be kept in check continuously, which also means that the post-mitotic cell, the myotubes, need to continuously confirm its decision to stay arrested, meaning it needs to continuously express P21 in order to remain arrested.
Was this experiment before Yamanaga's factor
Yes.
discovery and everything?
Yes.
Okay.
Why are you asking?
Because it's more shocking to know this before knowing all the stuff.
Right.
Maybe now people are more ready to.
Agreed.
Yeah.
And so, the other question you might ask, perhaps even in the discussion of this paper, I don't remember whether it is there or not, but you might speculate that because generally cells don't like to waste energy producing things they don't need, presumably these molecules are there for another purpose. They are repurposed by the cell to do something else. But that's speculation. The authors don't have evidence for that. So, this is interpretation of the results. Oh, and now I will show you additional results. Of course, the paper was much more complex than what I've shown you. I just presented the main experiments. I will show another piece of evidence and see what that suggests to you. So, what I've shown you so far was made using, the experiments were performed using an immortalized cell line, myoblast cells, and the myotubes derived from it. So the authors said, well, but we know that primary cells, that is cells freshly explanted from the animal, which better represent the actual state of muscle in vivo, have a tighter control of the cell cycle. So let's see whether removing P21 also activates primary myotubes. And the answer is not so much. Here is the answer. Control myotubes do not reactivate. Suppressing P21 only reactivates, say, 2 to 3% of the myotubes. So, in principle, the activity is there too, but the efficiency is much, much lower, an order of magnitude, more than an order of magnitude lower than in myotubes from the immortalized cell line. Then the author says, well, it is possible that other inhibitors come into play in primary cells. And so they suppress a number and a combination of those inhibitors. These are all inhibitors of the cell cycle, and when they're indicated, it means they have been suppressed. So if you suppress P21 plus another inhibitor called P18, you get more myotubes into the cell cycle. If you suppress P21 and P57, more. if you suppress P21 and P27, oh, this is 57. P21 and P27, you get even more myotubes into the cell cycle. If you suppress 3 inhibitors at the same time, even more myotubes. If you suppress 4, even more myotubes. All right. But interestingly, no matter how many inhibitors you suppress, if you don't suppress P21, you don't get any myotubes into the cell cycle. Now, these are the data. What are the results? What do you conclude from these data?
The P21
is necessary to
or
keep shut down.
Exactly.
or regulate the other inhibitor, the activity of the other inhibitor.
Let's stop there for a moment.
Yeah.
This is interesting too.
This is
Ah.
conclusion.
Exactly. The logical conclusion is that P21 is critical. It is the only inhibitor that really keeps the myotubes arrested. The others are somehow addition to P21.
But doesn't this also means that in vivo is a lot stranger because there are more breaks and probably more engine on. So doesn't
Yes.
make less sense than before.
Exactly. But now let's try. First, I encourage you to think in functional terms. Now I'll ask you to think in biochemical terms. That is, biochemically, how do you explain this result? How is it that P21 is so critical, and what's the role of the other inhibitors?
P21 è a monte.
Yeah, it is upstream, but that's a formal explanation. Biochemically, what does it mean? Where does P21 sit? What does it bind? How does it behave? That's the kind of explanation I'm asking you to come up with.
Because we know from the previous experiments where it biochemically it binds the complex, but this means that the P21 has other function besides that.
Why do you say so?
Because if the only function was
Remember, this graph is upside down, meaning when you see P21, it means P21 is being suppressed.
yeah. And it means that that complex is active if
Exactly.
P21 is suppressed.
So you have to explain why P21 is critical, and yet the other inhibitors play a role. So we have already accepted that there are complexes of cyclin D3, CDK4, and P21.
Yeah.
And if we remove P21, they are activated.
Yeah.
Where are the other inhibitors in this model? You have to put them in. Because
Yeah,
they're relevant.
they will
The
come
data
after.
show that they are relevant, so you have to put them into the model.
I think that the complex P21 and CDK go to active the other inhibitor or something like that.
So what you're thinking is that once the kinase is activated, it promotes the expression of the inhibitors. Is that what you're saying?
When the complex CDK bind the inhibitor P21,
Yeah.
then we have the activation of the other inhibitor.
But that would mean that if you remove P21, you will
Yeah, because
not
you
have the other.
because you have not the complex P21 with with CDK.
Alright, you no longer have P21 in complex with CDK4 and cyclin D3. And yet, the other inhibitors
Yeah.
appear to be still able, to some extent, to keep the myotubes arrested. not completely, but to a good extent.
Or maybe there is a correlation between the number of P1021 and the number of CDK or the complex that we have to inhibit.
Okay, so you think there are
The cell
more
can,
complexes?
yeah, more inhibitor we need, but I don't think so because the graphic show us that we have just, if we stop just P21 or P21 and P18, we have not so
Not so many reactive
Yeah.
myotubes. Right.
So,
So, again, I'll let's see. You have to make sense of these results, and you try to
And we
picture.
have to change the model. I don't know.
No, you don't have to change. You must. In this case, you should integrate the model with the new data. That is, these data are not incompatible with the previous model. Simply, you need to add extra elements into the model. That is, picture this. You have cycling CDK4 cycling D3 complexes with P21. Now we remove P21.
we
And
have,
yet, most of these complexes
Yeah,
are still
still,
inactive because if we remove P21,
Yeah.
very few myotubes are activated. So, what do the other inhibitors do?
At the same
The
as
same,
P21,
but,
yeah.
They replace P21.
Ah,
They do the
okay, it's
same.
just-
If you remove P21, they go there and attach more or less to the
Okay.
same place. They couldn't do it before because P21 was there. But now that you have removed P21, they come in
Ah,
okay.
okay.
and stop the kinase again. And then you remove that, and another one comes in, etc. That's why the number of reactivated myotubes keeps increasing while
Yeah.
the more inhibitors you remove.
Yeah, from this graph, like, how many are there? Because, like, even with blocking 4, it's just 35%, and in the last experiment, we have 70%. So
Exactly.
I can't.
There are 7 known inhibitors,
Okay.
and the authors only played with 4. So
Okay.
these results are still compatible with the possibility that the other 3 inhibitors are there and play their own role, or it is possible that primary myotubes have lower amounts of cyclin
Cyclin
D3 and CDK4. This is undetermined. This paper does not
Okay.
address that. There is a variety of possibility, but the simpler one, remember Occam's razor. The simplest explanation, at least the simplest that comes to my mind, is that simply the inhibitors replace one another. The more you remove, the more come in,
Yeah.
until they are exhausted, of course. And so based on the relative activity of these inhibitors, it appears that P27 is possibly one of the most powerful,
Yeah.
in that by simply adding P27 to P21, you go from 2 to about 18. So it's a large increase. The others probably, although it isn't entirely clear, we have to run more experiments, the others probably have a smaller role. But anyway, it is possible to make sense of this result, not only at the functional level. The functional level, we just say that they all concur in, they contribute to keep the myotubes arrested, although P21 is the primary responsible for the arrest. At the biochemical level, we have just devised a new model that says whenever I remove an inhibitor, another one comes in. And that's testable. The others didn't do it, but you can test it. So the idea depicted here is once you remove P21, the others come in and replace it.
And they know or someone study after this research if the complex P21 and CDK4 have I don't know, any activity, or is just going to degradation? So when
Oh, you mean other than driving the cell cycle,
Yeah.
another activity?
yeah,
Uh,
something like CDK4 can do inducing a cell cycle, or something else if the cell don't need it.
Some people have looked at the activity of cyclin D3 in myotubes, but they haven't come up with a real good answer to the question, what does it do? What they could show is that if you remove cyclin D3 during differentiation, or before differentiation takes place, then differentiation is not as good. It is somehow inhibited, suggesting that D3 is there to help differentiation.
Okay. I was asking just because we show before that it's constitutive activity. So
Didn't get it?
CDK is present all the time in the cell.
Uh-huh.
So I was thinking if we shift from one activity to another.
Uh-huh. Right. It is something that should be investigated indeed.
Okay.
But nobody, to my knowledge, nobody's done. All right, so because we have time, I'll show you another piece of information from this same paper. And this is very large, but don't worry. We'll just consider a few elements here. So the authors engage in a quantitative assessment of the various players in the game, or at least some of them. So remember this model here. They say, well, once again, the simplest explanation for P21's special role is that it is the most abundant inhibitor in the cell. So if it is the most abundant, it will be the first to block the kinase statistically, and only when you remove it, then the others come in, have a chance to come in. But that isn't the case. So let's see what the authors measured. All right, so here is myoblasts and myotubes. They have similar amounts of CDK4. They have larger amounts of P21 and larger amounts of P27, which is probably the second most important inhibitor after P27. And P18 as well is also drastically increased. But this is relative quantitation. So they know that these proteins increase, but they don't know what's the relationship between the various inhibitors. Is there more P18 than P21 or more P27 than P18? They don't know. They cannot possibly know from this Western, because Westerns are not absolutely quantitative unless you have a reference. And so the authors use references to quantitate absolutely P21 and P27 in myoblasts and myotubes. So now the results, the data, are expressed as picograms per microgram of total protein. So there is 2.5 picograms of P21 per microgram of total protein in myoblasts, and 2 or 3-fold more in myotubes, which is in good agreement with what we see here. But now we have absolute amounts. Now look at P27. It is 7-8-fold more in myotubes than in myoblasts. And also, in absolute terms, there is much more P27 than P21. Because they have similar molecular weights.
Yeah, I was looking up, like it's 17
Kilodalton.
Kilodalton, yes.
They're very...
So because they have similar molecular weights, I was saying we can compare weights roughly directly. Otherwise we would have to make molar calculations, which is more. All right, so the simplest explanation, Occam's razor, in this case, is not the right one. There is not an excess of P21 compared to P27. We don't know about the others, but already this explanation fails the test. So it is not confirmed by the test. Good, so other hypothesis. There is more P21, it has a higher affinity for the cyclin and CDK, so there is more P21 associated with the kinase and than P27. And perhaps the extra amount of P27 is somewhere else than something else, but does not inhibit. But not even that is correct, because here the authors quantitate the amount CDK4 associated, that is, they immunoprecipitate CDK4 and measure the amount of co-precipitating P21 and P27. In myoblasts, where P27 is tenfold more, and in myotubes, where it is perhaps eightfold more, but still much more in absolute terms. So, it's not even true that P27 is less associated to the complexes, and so
why is
why is P21 so important if P27 is more abundant and more bound to the complexes? And here the authors have no explanation, they don't address the problem, and so we can only speculate. And because we should speculate, we can say anything. There is no proof against what we're going to say. So try to explain why P21 is more important than P27, even though there is more of it. There is more of P27, clearly more of P27 associated with the complex.
maybe P27 need the presence of P21 to work, even if it's a lot present without P21. In some ways
Possibly, but there is no evidence for that. In fact, they compete for the same sites on the cyclin and CDK. So there are other competitors than synergistic molecules.
Probably P27 have another role in the cell.
Right, but still it's inhibiting, according
More.
to this, the majority of the complexes. And yet we remove P21 and at least some of them are activated. So, or what's more difficult to explain is that you need to
More.
remove P21
Yeah,
in
and
order
that's
to activate the
the-
myotubes. Because you might say, if I remove P21, only 2% of the myotubes are reactivated. So that's consistent with the fact that P21 is present in relatively low amounts.
but if you remove
But if you remove
P27,
none is
yeah.
activated. If you remove P27 plus two others, none is activated. So again, the simplest explanation doesn't work. We need a more complex, a more sophisticated one. What can we say? This is difficult. Not impossible. So there are various possible explanations.
It's useless looking at the data because they are clear and they don't
Exactly.
they don't help.
You have to come up with a model. Imagine those blocks that we were playing with in the graphical model. cannot bind together. We said
Yeah.
that they are competing with each other. You're talking now, we are confining our reasoning to P21 and P26.
Maybe P21 also binds something else that I don't know is needed to not to keep the differentiation.
Okay, so what you're proposing is that P21 is what we know about these complexes, but in fact there are other molecules that are there only if P21 is there,
Yeah.
preferably if P21 is there. And if you remove P21, they are also gone, and so the complexes are now active. That's a possibility. There is no evidence in the
Yeah,
literature for that, but
is no.
it's a formal possibility, yes.
Maybe we should use we used before the complex to to find P21. Maybe we should use P21 to find
Yes.
whatever
Yes.
else it
That's a
finds
possibility.
and look if there are
Absolutely.
other stuff.
A simpler possibility that does not need to introduce another
Other
hoc
variable,
explanation,
yeah.
a new variable that we don't know anything about, which you did, but it's sometimes it's necessary to make sense of results. We might say perhaps that there are different subpopulations of cyclin D3 CDK4 complexes. For example, you don't know that, but cyclins and CDKs shuttle from cytoplasm to the nucleus. So perhaps these
Where
complexes
it is,
here are in the cytoplasm where they would be,
Yeah.
it doesn't matter whether they're active or not because their substrates are in the nucleus. While these ones are in the nucleus, and if they get activated, then cell starts. And we could speculate more and think of other possibilities. It's fairly easy, but we'll stop here because there would be really wild speculation in the absence of data. And so we'll go to the final highlights of this lesson. Oh, finally, I translated this. So this refers to the whole process of the scientific project. So to start with, the questions must be focused so as to suggest strategies for finding answers. So in these particular cases we examined today, the authors focused on a particular aspect, if you wish, a detail of the whole system they are considering. The detail was, why do I need so much cyclin D3 and cyclin D1 and CDK4 to obtain a physiological activity of CDK4? Next, the questions, the experimental approaches, and their execution are often modified on the basis of the results obtained during a research project, which means oftentimes we have to go back and rethink what we have already thought about. In this particular case, for example, it's not the only example, the authors modify, in fact, throw away the original model and devise a new one to explain data they didn't know in the beginning. So they had to go back to the drawing board and draw a new model. And in fact, models, if clearly inconsistent with observations, as it was the case here, should be changed or abandoned without hesitation. And finally, interpretation of the results depends in large part on the point of view of the observer. So what the authors did, the engine is on and you need to keep the brakes down. It's an opinion, it's a point of view. There is no compelling evidence to say that that is so. It's an interpretation of the results. All right. So, because I somewhat expected, we would have some extra time at the end. If you wish, I can show you something else.
Of course. Yeah.
Okay. I brought with me another presentation. From the same authors, this is a different project, later project. Okay, the authors start from some facts that they found. That is, proliferation of terminal differentiated cells cannot be obtained. Why can it not be obtained? Well, the authors have devised several ways to reactivate the cell cycle in biotubes, such as expressing E1A, a viral protein that is able to reactivate the cell cycle. They can express cyclin E1 and CDK4 or 6, which is a single molecule. They can suppress the cell cycle inhibitors, cyclin-dependent kinase inhibitors. And all of these manipulations do reactivate the cell cycle in myotubes. Yet, the myotubes never really proliferate extensively. They do divide. They do increase in number for a short time and then they die off. If you use E1A, they die mostly by apoptosis. If you reactivate them by overexpressing cyclin E1 and CDK4, they usually arrest in G2, the first G2 after DNA replication. If you suppress the inhibitors of the cell cycle, you get mitotic catastrophe, so-called, which is a extreme breakage of chromosomes, small segregation, micronucleation, so a catastrophe, really, and apoptosis. So, the authors here were confronted with a variety of different outcomes, which depended on the particular means used to reactivate the myotubes. And so apparently, at first sight, the conclusion is doing different things, we obtain different results. None of the things we have done so far really reactivates the myotubes in the proper way, and so they die off.
But
That's it.
it isn't like a strong sentence to say cannot be attained. Like they tried a lot of methods, but
Thus far. Thus far.
it's
All right, I agree with that. All right, but the authors ask, is there a fundamental principle that explains all of these results? And they basically search for some commonalities between these different situations. And eventually they find one, and that is, the hint was due to the fact that they knew that by removing cyclin-dependent kinase inhibitors, they could not only reactivate plasmatoid myotubes, they could also reactivate reversibly quiescent cells and even senescent cells. All of these cell types are reactivated by removing cyclin-dependent kinase inhibitors. But there's a fundamental difference, that is, when you reactivate quiescent cells or senescent cells, they do indeed proliferate. Here is quiescent cells in the absence of growth factors, nonetheless double in number within 4 days. Control cells nearly don't grow at all. Senescent cells, you can reactivate them most efficiently by removing P21, but to some extent even removing P16. It depends on what particular inhibitor is most important in that particular type. Control cells are flat, they're senescent, they will not proliferate. And these cells triple in 3 days, so they really proliferate, and they're fine. Myotubes die. So that strongly suggests that it's not the means the authors are using to reactivate the cells that kill the myotubes. It's something intrinsic to the myotubes, right? What could it be? I'm not asking you. I'll tell you what it is. The authors noticed, among other things, they observed these cells, they studied these cells, and they at some point realized that when they reactivated fibroblasts, that is quiescent cells in this case, they proliferated and they had normal morphology. This is a mitotic figure, which is normal. But I've already told you that if you reactivate myotubes, you get mitotic catastrophes. This is a catastrophe. This is a nucleus. This is another nucleus. These are all mitotic nuclei, but they are fragmented. They are devastated. Okay, there are many ways to explain a mitotic catastrophe, but one way is by supposing that they enter mitosis without having completely replicated their DNA. And
there wasn't already an experiment about how many DNA
exactly.
okay, that you showed us.
Exactly. And indeed, that experiment stems directly from this observation. Another way to show that indeed myotubes do not completely replicate their DNA before entering mitosis is this. The authors have shown it in many ways, but the best pictorial way to show it is to stain one metaphasic nucleus of a reactivated myotube. It's a very ugly metaphase, but it is a metaphase. I think you can recognize it. It is ugly because it's a mitotic catastrophe. These myotubes have been incubated with BUDR throughout the cell cycle, that is, since their reactivation. So they have incorporated BUDR in whatever DNA has been replicated, and indeed they show BUDR signal in the nucleus. But if you now superimpose these two pictures, you clearly see that not all of the DNA has been replicated. Okay, so that, among other pieces of evidence, shows that myotubes can be reactivated, but never, in any circumstances the authors have tested, never completely replication. Now, the question, the model now is this. The authors say when we reactivate fibroblasts, they duplicate their DNA entirely and then proceed to mitosis. The sister chromosomes separate nicely, and that's it. When we reactivate postmitotic cells, say a myotube, DNA replication is incomplete, which I will spare you the molecular details, but the authors can explain why, depending on how the myotubes have been reactivated. Sometimes they go straight into apoptosis from aspase into apoptosis. Sometimes they proceed to G2 and rest there. Sometimes they even make into mitosis, but then what happens is that they cannot separate sister chromatids because they have not been completely replicated. Plus, they have badly fragmented DNA because, again, the replication is incomplete, and that causes catastrophe and eventually death. So, the question now is, why is it so? So, we have already concluded that it is not the experimental means that the authors use to reactivate the cells that causes this catastrophe. Or anyway, the inability to proliferate. Or, by now we can say, the inability to complete DNA replication. because other cell types, other non-postmitotic cell types, do replicate their DNA entirely. So there is something intrinsic to the myotubes that prevent them from replicating fully their DNA. And so the question is, what is the molecular nature of the obstacle to full DNA replication? This is the question the authors had in mind. How would you answer?
With an experiment.
Difficult one. Rather than answering, what would you suggest?
To show if the synthesis of DNA is for...
Before that, here we have a question,
Yeah.
which you realize is very difficult to answer. What is the molecular nature? What do I know?
I start to know what the...
What's the first rule we stated about questions?
That they have to be.
That you should focus them.
Yeah.
Yeah.
This is an unfocused question.
Yeah.
What is the molecular nature? God knows. It's not approachable,
Yeah.
put this way. So,
We have to find the approachable question.
In the myotubes and go in the myotubes and reformulate the question. Why in the myotubes the DNA cannot fully replicate?
What is the difference between myotubes and other cells that can be properly activated?
okay, so, first of all, your rephrasing the question is already a bit better because it focuses on the myotubes and on DNA replication in the myotubes. But then it asks why, which is a damn question in biology.
Okay.
Why is never a good question.
Okay.
You can, at the most, you can suggest a why after you have the results. Never start asking why. It's a loser.
Okay.
You were suggesting something.
Okay.
Well, what is the difference between myotubes and cells that can fully replicate their
Good.
DNA?
Good. That's a better question. And then
How do we approach?
how do we approach that question? Can we
Yeah.
focus it farther?
Because
Or does it already suggest an approach?
yeah, like an epigenomic approach, an epigenetic approach.
Okay, it could be some epigenetic marks, but
Yeah,
it
because-
could be even a huge number of other differences, right?
Yeah.
You are thinking of an epigenetic modification, which is certainly a possibility, but you don't really know. I'll stop you here because what I'm going to show you is not something that you're likely to guess. The way the authors proceed is by splitting this question, or a slightly better focused question, as Karim suggested, into
What stopped the DNA replication in myotubes?
yeah, and they say there may be two classes of obstacles to full DNA replication. One is functional. By a functional obstacle, the authors mean whatever is wrong in the myotubes concerning molecular biology, biochemistry, gene expression, molecular localization, molecular interactions, molecular modification, protein modification, whatever. So whatever is molecular biology and or biochemistry. Something that doesn't work the way it is supposed to, and so DNA replication takes place, but it is imperfect, and so it's never carried out to the end.
With this, didn't we like have everything like all the biology with this. What is like
It
another?
looks so at first sight.
What's the
So-
other one? Like a topology, some like shape? No, some like mathematical one. If it's not functional, it was physical, like
physical.
yeah, some physical one because functional.
Such as, how do you picture a physical obstacle?
like a wall, no, a wall.
Like a wall. Yeah.
I don't know, a physical, a spatial, maybe they don't have space to fully replicate, maybe they don't have like,
So we're not talking about specific hypotheses. Because
yeah,
it's
okay,
way too early to make specific hypotheses.
Yeah, but-
the original question is too ample. The authors say another class is structural obstacles. That is, there is nothing wrong in the biochemistry. There is nothing wrong in gene expression in these cells, except the chromatin of myotubes is such that it creates an obstacle, a physical obstacle, for the polymerase to proceed. And so it continues replicating DNA up to the point where it no longer can. Good. Yeah.
We know if in the myotubes there is a system to repair DNA.
Yeah.
We have it.
Myotubes do have DNA repair systems. Not all of the repair systems that are present in proliferating cells. So, yeah.
Just to focus better the question.
So, I won't go into the functional part. There is nothing extraordinary that the authors or anybody else in the world have found in the myotubes that would explain why they cannot replicate their DNA in full. But the authors, and after all, this is the obvious explanation. There must be something wrong, but it's difficult to find what. This is more attractive because it's simpler, it's more elegant as an explanation. It's more straightforward.
yeah, that's what I thought with epigenetics, yeah, some
Yeah.
change but it's not so approachable, like,
It is difficult to find an approach, but you can study chromatin structure, can you not?
the difference from the structure of but we have to look at where it stops, where is the
Where is the arrest? That's
Yeah.
a good question. That's approachable,
okay,
right?
Yeah.
Good. So, you see, during the discussion, we go over the evidence, we go over our own reasoning, we come up with better and better idea until we find an approachable question. Where is the arrest? Where is the stop? That's an excellent question, because once you identify where DNA synthesis arrests, there you look for the obstacle. Clearly focuses you onto something that is hopefully approachable. Good, so what the authors did to identify the obstacle was to reactivate the cell cycle myotubes, induce DNA synthesis, label DNA with UDR, and then using a different, an old technique, no longer in use in most laboratories, but in this case, for reasons I don't want to explain because it will be too long, they resorted to separation on a cesium chloride gradient, which exploits the different density, the different weight of BUDR substituted DNA compared with the regular DNA. Because BUDR contains bromine, which is a very heavy atom, then that DNA is heavier and separates upon ultracentrifugation in appropriate conditions. So the authors take BUDR substituted DNA, which in these conditions is the only DNA that has been synthesized. And they hybridize it with genomic chips. So each point in these chips represents a different region in the genome, and what they're looking for is not where DNA hybridizes, because
But
where
where
it's been
it's not.
replicated. They're looking for spots in which it does not hybridize. There's no DNA there because there has been no replication. Wonderful, the perfect approach, except the result is this.
So it's random.
Yes,
It
you
blocks
got it.
in random.
It's random. So you see, this is these experiments are rather noisy, meaning that in the same position you have points in many different positions, in many different places, positive or negative. But basically, without going into the details, if you smooth out the curve, that is, you average
Yeah.
the values in several regions, one next to the other, this is what you get, a line that oscillates around 0, which means, translated in practice, that in each single nucleus, every region
There is
has
the
the
same
same probability
to
of being
stop.
replicated or not. There is no difference anywhere. This is very disappointing because after this experiment, the authors found nothing. They have not found a region in which to look for the obstacle. But they don't throw away the results. They think twice about the results because these data were also telling something about an unexpected truth. Do they suggest anything? The fact that it is entirely random whether any region will be replicated or not before DNA replication simply arrests, stops. It might or might not suggest you, depending on whether you know about a specific biological phenomenon, which is called replication timing. You don't know. Most students don't. Replication timing is that phenomenon that you might have heard about once I explain what it is. According to which not all DNA is replicated at the same time. There are regions in the genome that are replicated early in S phase, others that are replicated in middle S phase.
So this is strange.
So this is strange. Exactly. Because
The probability shouldn't be the same for everyone. It should be
if
more.
they stop at some point during S phase, you would expect only early regions to be replicated, not late ones. The fact that there is no difference among the regions means that in these myotubes, the DNA is replicated all together.
Yeah.
Then sometimes it fails, but it is still, the polymerase is attempting to replicate it all together without differentiating between early and late regions. Now, the authors decided to go after this result because it's very peculiar and it is potentially very useful. Because replication timing at the time of this experiment had been described in sophisticated details, but had not been explained at the molecular level. One of the reasons because it had not been is because replication timing is basically universal in nature. All cells, except very early embryo cells, replicate their DNA with a specific replication time, which is typical of every cell type. Each cell type has a typical replication time. In fact, it's like a fingerprint of a cell type. The fact that you don't have, in order to study a phenomenon, usually you want to have it or not have it to see what makes what difference it makes. But you don't have that. You don't have that situation, except in this case,
Now
the
you
authors now have it. They have a situation in which myotubes replicate without a replication timing, and they have another situation. Here is what you get in myoblasts. They have replication timing. And okay, here it is. This is another way. This is now done not with chip hybridization, but with next generation sequencing. But it's basically the same. Early regions are in red, late regions are in blue. Myoblasts have a defined replication timing. CDKI inhibitor depleted myotubes have basically no replication timing. But myotubes reacting with UNA do show replication timing, which is a little shorter in amplitude, but still replicates faithfully that of myoblasts. So one conclusion might be that they remember they have been myoblasts and they follow the same replication pattern. But what's important is that now the authors have a system, the myotube, which can replicate DNA in the presence or in the absence of replication time. That's a great system to address the molecular basis of replication time. Now, that's a lesson in treasuring results that apparently have failed. So the original question was not answered by the results, and yet the results suggesting a very interesting new approach, new possibility, new line of research. But let's go back to the original question. What is the molecular nature of the obstacle to full DNA replication? I'll be very short. One question before this, before asking what is the structural obstacle, is there a structural obstacle, or is just a hypothesis of ours which has no basis? So, in order to determine whether there is a structural obstacle, the authors come up with an idea. What would you do to determine if there is a structural obstacle? In principle, I don't want the experimental details that you don't know. But how would you address the question if there is or not a structural obstacle to DNA replication?
Simplifying the structure,
That's
linearized, and, but it will.
Induce add a DNA part that I know and show if the replication system of the myotubes go to replicate my DNA.
Not bad, not bad. In that way it cannot be done, but it can be done in a similar way.
Okay.
So I'll show how the authors have done this experiment, how they address this question. And it's basically what you propose, except it is not easy. That cannot be done in vivo, or at least it's. It can be done even in vivo, in fact. Now that you mention it, but the authors chose to do DNA replication in vitro.
Okay.
Now, there is basically one widely used system for in vitro DNA replication, which is using Xenopus frog egg extract. The eggs, basically an extract is lysing the oocytes, the eggs, spinning down, centrifuging down whatever is not soluble, and take the supernatant. That's the extract. That extract contains everything that's necessary to replicate DNA. In fact, you can put even a naked piece of DNA into it and it will replicate. And you can put a full nucleus into it and it will replicate. So,
Oocyte of what? I didn't.
Frog.
It's a Xenopus levis. Xenopus levis.
Okay.
A well-known frog because it's one of the much studied animal models of which we will hear in the next lesson. Okay, so the authors reason this way. They isolate the nuclei from non-reactive myotubes, wild-type straight quiescent myotubes, and they put them, incubate them into Xenopus egg extract with a label that's used to determine whether DNA is replicated. Yeah.
The myotube had the inhibition of P21.
Yes, they were not reactivated, not manipulated.
Oh,
just
okay.
Unperturbed myotubes. So there are two possible outcomes of this experiment. Either the nuclei are completely replicated,
So no structural.
so there aren't probably any structural problems. Simply the myotubes are missing some component that the authors haven't found yet, and yet when you put them in the proper conditions they can be replicated. Or you still get partial DNA synthesis even in the extract, and that clearly shows that there is a structural problem because we know that all the components necessary to fully replicate DNA are there. And the result of this experiment is that if you take nuclei from myoblasts and myotubes, here are the myoblasts, replication, and these are the myotubes. So clearly they show evidence of a structural. I'll stop here because it's late. And these results are not finished yet, so I can't tell you what's the final.
No, I want to know.
I wish I knew too.